Welcome to the deep dive. Today we're tackling a really pivotal question in pharmaceutical development.
hmm
How are parallel synthesis and automated reactor systems changing the game? Basically, how fast can we create new medicines now?
It's a critical area, definitely.
So you, our listener, are going to get a clear understanding of how these advanced tools and also things like small-scale experiments, computer control,
Statistical design, too.
right, and statistical design, how all that is driving efficiency, getting therapies to patients faster.
Yeah, and you might have noticed things like the special features section, new technologies and process research. There's real interest here.
Exactly. So our focus today is pretty sharp. How do these approaches actually optimize the development process and improve scalability?
And ultimately shrink that timeline for new drugs.
Okay. So where do we start? Parallel synthesis.
Let's. It's fascinating how these technologies sort of converge.
Mhm.
Parallel synthesis, at its heart, it just means making lots of different compounds at the same time.
Instead of one after the other.
Precisely.
Mm.
Not that old linear way. Imagine generating whole libraries of molecules side by side.
And then you combine that with automated reactors.
Yes.
Yes.
And these are often small systems, our sources mentioned, like
Yeah.
10 to 100 milliliter scales. Tiny, really.
Wow. Okay.
But they give you incredibly precise control. You can monitor lots of different reactions happening all at once.
Okay, let's dig a bit deeper there. So moving from sequential to parallel, what's the immediate hit for someone trying to find a new drug?
Well, the biggest immediate impact is speed, a dramatic acceleration, especially in those early discovery stages.
Right.
You can suddenly screen a much broader range of conditions. Try different catalysts, temperatures, solvents,
All at once.
all at once. So the scope of your experimentation just explodes. And you get a richer understanding of the chemistry involved in way less time.
So it's not just saving lab hours, it's
Yeah.
potentially changing what's even possible.
Absolutely. It can alter the economics, make research that was maybe too slow or expensive suddenly viable.
That makes sense. Now, we know early drug development often, well, it throws up some tricky challenges, doesn't it?
Always.
Like one source mentioned poor water solubility. Lots of promising candidates have that issue. How do these parallel techniques help there?
That's a great example
Mm-hmm.
because you can quickly make and test lots of variations, analogs of your lead compound.
Tweaking the structure slightly each time.
Exactly. You systematically explore changes designed specifically to improve properties like solubility. Make a small change, test it quickly, see the impact. You navigate that chemical space much more efficiently.
So it's like this engine for exploring possibilities around a starting point, trying to find that sweet spot, the right properties for a drug.
That's a good way to put it. An efficient engine for molecular exploration.
And you mentioned working small scale. Why is that so important, the miniaturization?
Oh, it's huge for efficiency. Resource efficiency. You need way less starting material. You generate minimal waste.
Which lets you do more experiments.
Far more, yeah.
Yeah.
And critically, the data you get from these small parallel reactors, it can actually be quite representative of what happens on a larger scale.
Really?
Yeah, one of the sources really emphasizes that. So you gain crucial insights without the massive cost and material needs of big trials.
Interesting. So insights from tiny experiments can actually, guide the scale-up process. Are there limits? Pitfalls, maybe?
That's a very important question. It's not always a perfect one-to-one translation, of course.
Okay.
Things like, say, heat transfer or mixing behave differently in a huge vat versus a tiny vial. That's physics.
Right.
But having that fundamental understanding early on, optimal conditions, potential impurities, reaction kinetics, it's incredibly valuable.
Right.
It means you make much more informed decisions when you do scale up.
So you avoid major surprises later, less costly rework?
Exactly. You reduce the likelihood of hitting big problems. Think about that example from one of the sources. Synthesizing over 500 quinoxylene derivatives.
For anti-TB drugs, yeah.
Right. You just couldn't explore that much chemical space practically or affordably without doing it small scale in parallel.
500, wow. That really hammers home the point. And didn't that same source mention modifying exocyclic groups?
Yes, for fighting drug-resistant TB.
Where even tiny structural changes had big effects on activity?
Precisely. Like the position of one methyl group. That level of fine-tuning is perfect for these automated parallel setups. You make the tweak, you test it fast.
A very efficient way to optimize how the molecule hits its target.
You got it.
Okay, let's shift to the computers. The control and automation side. It's not just doing many things at once, it's the way they're done, right? The precision.
Absolutely critical. Computer control gives you incredibly accurate handling of all the key parameters: temperature, stirring speed, how fast you add reagents.
Very tight control.
Very tight, which leads to much more consistent, reproducible experiments compared to, well, doing it all by hand.
And that consistency must be vital for trusting the data you get.
Oh, absolutely. Plus, these systems usually log data automatically.
Ah.
They monitor progress,
Wow.
yield, impurities, generating this huge amount of data that's instantly stored and ready for analysis. Much easier than scribbling in a lab notebook and trying to spot trends later.
So speed, efficiency, tons of high quality, consistent data. But how do you decide which experiments to run? Out of all the possibilities, that's where statistical design comes in, DOE.
Yes, you've hit on a really key piece. Design of experiments, DOE. It gives you a smart strategy for planning these parallel runs.
So not just random trial and error.
No, no. It's systematic. DOE lets you vary multiple factors, temperature, concentration, time, whatever, all at once, but according to a specific efficient plan.
Okay, so instead of changing just one thing, seeing what happens, changing another, you adjust several things together systematically. What's the big win there?
The win is efficiency and insight. By varying factors together, DOE helps you pinpoint the critical parameters, the ones that really drive the reaction outcome.
The important levers to pull.
Right. And crucially, how those parameters interact with each other, which you often miss with a one-factor-at-a-time approach.
Interactions. Okay.
So you get a much deeper, more comprehensive understanding of the process, but with significantly fewer actual experiments. It adds statistical rigor.
And that rigor helps build a robust process, something reliable.
Exactly. Because robustness is what you need for manufacturing, isn't it?
Yeah.
You want a process that works consistently, even with the small variations you inevitably get in a real plant.
Makes sense.
So DOE helps you find not just the best conditions, highest yield, best purity, but also the acceptable range around those conditions, the operating window.
So you know how much wiggle room you have.
Precisely. And it also helps you understand and quantify variability in the process itself, which is key for ensuring consistent quality batch after batch. It connects back to needing consistency for reliable results, like in clinical trials. A well-understood process gives predictable outcomes.
Okay, so let's tie it all together. We have parallel synthesis, automated reactors, small-scale work, computer control, statistical design. How does this whole package speed up getting a drug from the lab bench to, well, to patients?
Well, all that knowledge you gain early on from those small, controlled, statistically designed experiments, it directly fuels process optimization.
You figure out the best way to make it much earlier.
Yes, you identify efficient, robust conditions right at the start. So when it's time to scale up for manufacturing,
Fewer nasty surprises, less troubleshooting.
much less, ideally. Early optimization heads off many problems that might only show up at large scale. Some sources talk about the challenges of scale up, and this approach really tackles that proactively. And fundamentally, just being able to identify those optimal conditions so much faster drastically cuts down the overall development timeline. Moving from discovery to clinical trials can happen much more quickly. The small-scale insights really do inform and streamline those bigger pilot plant runs.
Okay, so to sum it up then, key takeaways on how these technologies accelerate development.
I'd say, number one, a seriously compressed timeline because you explore and optimize so rapidly. Two, more efficient process optimization, leading to robust, reliable manufacturing methods.
And three,
And three, improved scalability. That transition from lab bench to factory floor becomes much smoother, much faster.
ultimately it sounds like these tools are directly tackling that urgent need, getting new treatments out there sooner.
Absolutely.
Yeah.
And with innovation constantly happening, like those new technology sections highlight, we can only expect these tools to get better and have an even bigger impact.
Yeah. So as you, our listener, think about all this, it really makes you wonder, doesn't it? How will pharma development keep evolving as these technologies get even smarter, even more widespread? What else might they unlock down the road? It's definitely a space to watch. Thanks for joining us for this deep dive.